this opportunity to share some of our ongoing research with you today. I guess in front of this audience, I don't really need to explain why regulatory networks and mapping them can be so important. However, I want to sort of go over the fact that one of the major bottlenecks in really applying regulatory networks to multiple systems is the fact that the current experimental methods for building regulatory networks are really limited. And mainly they're limited by the fact that it takes several hundred experiments just to create one regulatory network for one cell type. And if we want to start moving into regulatory network dynamics, we need to have a system that's much more scalable across multiple cell types and organisms. So really, we need something that can map many transcription factors at once and that can be readily applied to multiple organisms. And today I'm going to be arguing to you that we can utilize DNase 1 footprint maps as this approach, as we can map all DNA binding elements within the genome in one single experiment. So I know this has been discussed several times so far, but I'll give you a really brief overview of what exactly DNase 1 footprinting is. So pretty much what we're doing is we're leveraging the fact that within the nucleus, sites which are actually bound by transcription factors are highly accessible. Consequently, if I were to take a nucleus and just treat it with the limiting concentration of the non-specific nuclease DNase 1, it's going to cause cleavages within these accessible regions. Consequently, if I isolate these small DNA fragments which are released with this method and sequence the ends, those ends directly correspond to DNase 1 cleavages, and if I map them along the genome, you can identify sites which are hypersensitive to DNase 1 cleavage. These are DNase 1 hypersensitive sites, and they mark sites which are occupied by transcription factors within that cell type. And this is just a map across multiple cell types, where you can see sort of DHSs which are common in multiple cell types, as well as ones which are cell type selective. On average, you see around 100,000 to 250,000 DHSs per cell type. And also, a large amount of this data is publicly available through these two, the REMC as well as the ENCODE project, and it was recently published. However, if you sequence enough reads, you don't just identify which sites in the genome are accessible, but you can actually identify the specific nucleotides which are bound by transcription factors within those sites. So let me briefly demonstrate how this works using this DNase 1 hypersensitive site as an example. So if I actually zoom in to the individual nucleotide level, you can see that within this DHS, the cleavage rate is actually not uniform. Rather, the punctuated binding of a transcription factor right here occludes the bound DNA from cleavage by DNase 1, demarcating a footprint which marks the transcription factor occupancy at nucleotide resolution. You can see that this footprinted region is conserved, indicating its functional significance. And if you actually zoom in on the individual DNA sequence, the sequence recognition motif for NRF1 can be readily visualized. And this can be further validated, or NRF1's occupancy can be further validated using ChIP-seq on NRF1, these exact same cell types. So if you look at NRF1 sequence elements within open chromatin in a cell type, what you can see is that these sites up here, which give the cleanest footprints, are also the most highly conserved sites. And furthermore, if you compare these sites which have nice footprints to ChIP-seq data, you can see that 89% of these NRF1 footprints are actually captured by ChIP-seq data sets within the same cell type, sort of demonstrating the ability of DNase 1 without doing an additional ChIP-seq to really capture the occupancy landscape of this transcription factor. And we've demonstrated this for a number of other transcription factors, which was recently published. Overall, we've applied this method to over 40 cell types, 41 of which have been published, and many others are currently available through the REMC and ENCODE websites. And we've been able to map 45 million transcription factor footprints across these diverse cell types. And furthermore, by combining these with transcription factor motifs, which are currently within these databases and are only getting better by the month, we can really highlight which transcription factors are potentially occupying those sites within a given cell type. So how can we utilize this to actually build a network? So it's pretty straightforward. What our approach is to take a transcription factor promoter gene, scan within the 5 kb surrounding the transcriptional start site, and just ask which footprinted binding elements are present within that cell type. So say within Th1 cells here, the IRF1 promoter has these footprinted elements. Then you can further go and ask, okay, where did IRF1 bind? And you can just generate sort of a small network with this and just repeat it for all 475 transcription factors in which we actually have these good motif data for. So this is sort of showing you can just now go on to IRF7, map it, and so on and so forth. So when you do this, you get a network, a nice hairball, as was introduced previously, that looks like this, and you can apply it across multiple cell types. In this study, which I'll be talking about today, we did it across 41 cell types. But two major questions arise. First of all, how accurate is this? And second of all, can you really inform biology by sort of looking at these transcription factor interactions in this network context? So to address the first question in terms of accuracy, we just decided to compare some of our networks to sort of known gold standard networks. So unfortunately, there's really not sort of these very large gold standard networks. We're really limited to comparing to these small subnetworks. So this is a subnetwork that was constructed using ChIP-qPCR in mouse embryonic stem cells, and we just wanted to compare how does this look to the network which we found in human embryonic stem cells, and you can see that they're quite similar. Similarly, this is a network which was constructed using perturbation studies, DNase 1 footprinting, as well as, I believe, some ChIP studies across multiple papers in developmental skeletal muscle. And we can see that within our myoblasts, we see a very similar network. However, the first question could be, oh, great, you're matching it, but you could be matching this network in every cell type, irrespective of whether or not that network should be in there. So to quantify that, what we can just do is just calculate, okay, if I were to generate this subnetwork in not just ES cells, but in another cell type, how close does it actually resemble this mouse ES cell network? And if you do that, you can see that this network is best matched in embryonic stem cells and is really poorly matched in all other cell types, sort of indicating the specificity of these networks in terms of their cell type origin. And similarly, if we do this with this network, we get a very similar outcome, seeing that this network is best matched in skeletal myoblasts and skeletal muscle. So now that we've mapped these networks, we now started wanting to look at the dynamics of these networks and how they change between different cell types. And to start, I just want to look at 8 transcription factors. These 4 pluripotency factors, which I previously introduced, as well as 4 hematopoietic factors. So if we just look at the interactions between these factors in ES cells, you can see that the pluripotency factors are really interconnected, whereas the hematopoietic factors show very few, if any, connections. We can do this across multiple cell types, and you can see, say, within myoblasts, you see very few interactions, which you would expect to not see that many. And furthermore, you can just color this based on which ones are specific to that cell type versus which ones are common. And you can see that pluripotency factor interactions highlight in embryonic stem cells, as to be hoped, and also hematopoietic factor interactions are highlighted in hematopoietic cells, sort of indicating the specificity of these networks and able to sort of inform in terms of which cell types these transcription factors are active in. Also, I just really want to highlight the diversity between these networks. Really, only 5% of the edges which we're calling are actually shared across all 41 cell types. Most of these edges are actually unique to one cell type, and there's a strong diversity between transcription factor networks in humans. So now what we want to do is just ask, okay, can we, given this diversity, can we identify cell types which actually have similar regulatory networks? And to do this, we wanted to focus on the transcription factors which are driving rather than just the individual edges. So we kind of took a simple approach by just asking, okay, let's just count how connected a given transcription factor is within that network and just see how that varies between the different cell types. So in this sort of subnetwork, you have IRF7 would have a total degree of 7, meaning it has 7 edges. And you can do this for many other transcription factors and get the actual count, which gives you a matrix that looks like this, which is 475 long and 41 cell types deep. You can then cluster this matrix, and what you end up getting is something that looks like this. Which is kind of striking here is the fact that you can see that similar cell or functionally related cell types actually have similar underlying core regulatory networks in terms of the transcription factors that they're utilizing for these networks. Such as you see the fetal cells and tissue clustered together, as well as epithelial and stroma. However, since we analyze this from a transcription factor-centric point of view, we can now actually pull out what specific transcription factors are doing. So let me demonstrate what I mean by that. So in this situation, we can see that actually OCT4 is showing up mainly in ES cells, which is what you would hope given the fact that this is a pluripotency factor. You see that it's mostly contributing to that network. Furthermore, you can look at other transcription factors such as GATA4, which is a known cardiac regulator, which you see showing up in fetal heart as well as great vessel derivatives. PAX5, which is a known lymphocyte regulator, shown up in lymphoblastoid cells as well as lymphocytes. And you can look at a number of different transcription factors, and really what you are seeing with this is the ability to pull out known regulators of cell identity as well as novel regulators of cell identity, such as HOXD9 in endothelial cells or HOXB1 in ES cells, fetal lung, or GATA5, which in fetal lung, which is known to have been a marker for lung cells, but not necessarily been known what it's doing. So now that we've looked at that, we want to sort of focus on not necessarily the transcription factors which are sort of changing their overall connections, but can we see anything unique about transcription factors which are expressed in almost all cells? But are they behaving differently within each cell type? So to do this, we actually focus on one specific transcription factor, retinoic acid receptor alpha, which is involved in numerous biological and developmental processes. And we wanted to ask, how is this transcription factor connected within the network in these different cell types? And to quantify that, we specifically focus on feed-forward loops, which are a network motif which is very common throughout biology, in which information is propagated forward from sort of a driver position down into these passenger nodes. So what I'm displaying, or I'm going to be displaying here, is I'm just going to quantify within a given cell type, which I'll put this first one up here, how many times do you find retinoic acid receptor alpha in the first position, the second position of the feed-forward loop, or the third position? If you do this across all 41 cell types, you can see that by and large, retinoic acid receptor is actually present within these passenger positions of the feed-forward loop. However, within blood and endothelial cells, you see this shift from being a passenger to a driver. And actually of note, when in acute promyelocytic leukemia cells, you see it shifting to become a very strong driver of the network. And furthermore, I really want to highlight the fact that acute promyelocytic leukemia is driven by a retinoic acid receptor alpha translocation. And this is really highlighting the fact that not only is this sort of messing with the downstream genes in terms of misregulating downstream genes, but this translocation product is also messing with the network itself, which you can see here. It's now shifting its role within the larger transcriptional regulatory network. And also, I just really want to highlight the fact that this is derived sort of without use of any expression data, but just with a single experimental paradigm, just given enough sort of normal cell types to compare against. So now that we've sort of looked at the dynamics of these regulatory networks, we now want to actually sort of look at the architecture of the networks. But before I go into that, I really want to give you an overview in terms of how we measure architecture and what's sort of been done previously regarding the measure of network architecture. So in terms of multicellular networks, there's really only one gold standard that exists. And this is the neuronal synaptic connection network that was produced by Sidney Brenner in the 1980s. And it's a beautiful paper of just electrographs after electrographs, really marking the individual connections, synaptic connections within the brain. And what was actually noticed in 2004 by Yuri Alon's group was the fact that if you look at this network, it has a very characteristic architecture which resembles other information processing networks within biology. So specifically, what they do. So networks are formed up of a set of building blocks. So these are, as I showed you earlier, these three network motifs. Feed-forward loops are one, as well as many others, such as regulated and regulated neutral. So what they just asked is, do you find any of these motifs within a network more often or less often than you would in random, using this normalized Z-score? For example, feed-forward loops within this network are found more often than random. And if you do this for all 13 network motifs, you get a snapshot of sort of the building blocks of that network. In this situation, you see that the feed-forward, the regulated and regulated neutral, as well as the clique and semi-clique are actually enriched, whereas these network motifs are actually depleted. So first, as a sanity check, we just want to see, okay, what happens if we don't use DNase 1 footprints to build these networks, but we just build these networks based on motifs alone, ignorant of anything else that's going on in biology, just saying, okay, let's just use the DNA sequence alone. And when you do that, what you see is that that does a very bad job. Or at least doesn't necessarily do a very bad job, but the network you get does not resemble this sort of common architecture of multicellular processes. So now if we actually go back and utilize the DNase 1 footprints, which is all the data I've been talking about so far, what you can see is that this is just for one cell type. It very nicely replicates it, sort of this conserved architecture. And furthermore, you can do this for all 41 cell types. And what's sort of really startling here is the fact that these networks are very diverse. They're, like I said earlier, only 5% of the edges are actually shared across all the networks, yet somehow they're all converging on this common architecture that resembles how a C. elegans brain works. However, one of the sort of questions was actually the result of this kind of surprised me. I just wanted to see, I was playing around today, I was like, okay, what if I make a super network and just combine all these 41 cell types into one cell type and say, okay, we have a great new cell type. What actually happens? What you can see is it actually does a poor job. It actually doesn't stay with the same architecture, but actually the architecture gets worse. So it's just sort of showing the importance of using cell type-specific regulatory networks. However, we don't just have data on humans. We also have data on mice from 25 different mouse cell types. So we can ask the same question. Does the mouse regulatory network sort of have a similar architecture to human? The answer is yes. And furthermore, we don't just have data on mammalian systems, but we also actually end up having some data on plants. And what we can see here is that actually, shockingly enough, the Arabidopsis has a very similar architecture to humans and mice. So showing that this is not just, as was known before, sort of a common thing of animals, but also this appears to be a common architecture of all eukaryotic systems, or at least ones which we've analyzed so far, showing this goes for both plants and animals. So with that, I think the major point I'd like you to take away is the fact that we can utilize these genomic DNase 1 footprint maps to provide a general scalable solution for mapping transcriptional regulatory networks that can be applied across multiple different cell types and species. And that usually using these maps, you can really gain insight into the underlying biology. And with that, I also just want to make a plug for a website which has all this data on it that you can sort of play around with your favorite factors and see how they interact within different cell types. So it was produced by Alex Reynolds in the lab. And with that, I'd like to thank all the people in my lab, or not my lab, obviously, all the people in John's lab, including Shane, who I worked tirelessly on this project with, as well as Sana and Christina for the Arabidopsis samples, and Elhanan, who was very sort of insightful when producing these network sort of topology and other issues, or fun stuff with that. So thank you. Luke?
Yeah, very nice talk, thanks. Yeah, I was just wondering if you can comment on your thoughts on if you plan to take this further, maybe to link binding sites to genes in a more sophisticated way than just the window, because it's not always the closest gene and so on, as we've seen in several talks on enhancers and so on.
Yeah, I completely agree with that sort of sentiment, the fact that we need to move beyond the 5 kb around it. And I think that we are getting towards the ability to do that. In one of the one of the papers which we had in Nature alongside the ENCODE paper, showing that we can link these distal elements to promoters using correlation studies if we have enough cell types. So I'd be utilizing the DNase 1 framework to still do it. Or so you can also just use an additional experiment on top of it using a 5C approach, or potentially a high C if you sequence enough reads, although it's a lot of reads at current rate, to link these. And I think that's definitely a needed thing to do. But it's difficult, is the answer.
Thanks.
So we have time for one more question. So I would choose one at the back, and maybe you can follow up later with.
So are you identifying which transcription factors bound at the location of the footprint based on DNA sequence?
Yeah, we're using DNA sequences. So in that regards, we're limited based on sort of our current transcription factor motif models. But nonetheless, I'm sort of hoping that, at least given the talks we saw on Monday and Tuesday, that with better models we'll be much better able to sort of nail down which transcription factor motif that is at any given site.
Can you tell apart, for example, when you have members of the same structural family which like to bind similar motifs, can you tell apart which member is bound there?
Given current positional independent matrices, it's very difficult. However, within this network, it ends up sort of clean itself up incidentally because of the fact we're utilizing both in degree as well as out degree when we're analyzing this. And say if a transcription factor is not necessarily expressed within that network, it will not have a good in degree because there actually won't be that many transcription factors regulating the promoter of it because of the fact that that promoter is just not hypersensitive because it's not actually active. But yeah, telling apart sort of similar motifs is currently difficult, but I'm really optimistic that within the next year or so we'll get much better ways of differentiating that using structural features or higher resolution binding elements. So,
Thanks.
Thank you.
Okay, let's thank the speaker again.